Welcome everyone, and thank you for joining us for our webinar titled Single-Cell Electrophysiology and Two-Photon Imaging in Awake Mice with 2D Locomotion Tracking. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This session is sponsored by Neurotar and will feature 3 lab groups presenting methodology and research findings from neurophysiological studies in awake, head-fixed mice. And now just a quick introduction to Neurotar's mobile HomeCage, which enables this research by providing the stability really required for electrophysiology and two-photon imaging, alongside an airlifted platform that allows the mouse to move around its environment. The clamp rotates to allow access to lateral parts of the brain, and special head plate design makes the head fixation procedure easy and ensures excellent imaging stability. The mobile HomeCage can be equipped with a built-in locomotion tracking device, which is fast, precise, and unaffected by ambient light. The tracking software is open source and can control external stimulation devices such as liquid reward during closed-loop experiments. More complex behavioral readouts can be implemented in the mobile HomeCage Large, for example, maze navigation or sociability studies. 98 labs worldwide have adopted this technology, and we have 3 of them here with us today.
Thanks, Liam. Hello, everyone. And John and I are here from the University of Bristol, where we work in the lab of Professor Jack Meller. And our main focus is looking at the mechanisms underpinning synaptic plasticity. And we're particularly interested in combining behavioral assays with in vivo electrophysiological recordings so that we can investigate the patterns of activity that induce plasticity during spatial navigation. So I'm going to talk first about our experiences using Neurotar's mobile HomeCage to track reward learning behaviours in head-fixed mice. And then John's going to speak a bit more about how we've been obtaining recordings from CA1 Pyramidal Neurons so that we can observe the membrane dynamics underlying plasticity during spatial navigation. So the most important factor in obtaining these intracellular recordings in an awake animal is the mechanical stability of the recording electrode within the brain, and this of course requires head fixation. And there are a number of existing methods to do this, each come with their own limitations, whether that's having to physically restrain the animal, so it kind of restricts the number of behaviours that you can measure, or in this instance where the mouse navigates around a virtual reality environment while running on a spherical ball. The animal isn't provided with any tactile sensory stimuli, which we know they naturally rely on much more than visual input. Or in some cases, you see that people use linear treadmills where it's possible to attach tactile cues. However, in this instance, you're limited to a single linear track, and so you're not able to look at movements in 2 dimensions. So we're very interested in using this mobile HomeCage setup provided by Neurotar because it allows you to head fix a mouse over a floating flat-floored arena which can be voluntarily moved by the animal, allowing them to freely explore the environment like this. And the mouse is fixed in the same location relative to an external observer, whilst maintaining the perception of moving around a maze. So here you can see how this looks from above. The mouse is fixed in this position here, and the whole environment moves around them. So this system allows you to track the animal's behaviour in 2 dimensions, whilst providing them with more naturalistic sensory stimuli that you wouldn't get in a virtual reality system. So we use a fairly flexible timeline for our studies, depending on the kind of behavioural task we're using. But in general, we surgically implant the head plate when the mice are about 7 to 8 weeks old. And we've previously kept mice for up to about 3 months like this without any issues, which is great if you have a long behavioural training protocol. And the mice are housed in pairs with plenty of cage enrichment and have several days of handling before being head fixed for the first time. And so for behavioural training, we use a sucrose solution delivered via a lick port. So you can just see here, this is the lick port here. The animal is delivered a reward and it's able to lick the sucrose. And we motivate the animals using a water restriction protocol whereby the animal is deprived of water overnight and then it's returned 1 hour following behavioural training the following day. And you can see from their body weights here that the animals lose no more than about 10% of their body weight each session and still show normal growth over time. And then here I've just got a measure of their activity in the first few sessions that they're head fixed. This just shows the number of laps that they run in about 10 minutes. And you can see that this stabilizes quite nicely after just a couple of sessions. And then finally, we create a cranial window 24 hours before the recording session so that the animal can fully recover from the surgery before having to perform a behavioral task. So next, I wanted to touch on a couple of ways that we've tried to improve the behavior of the mice while they're head fixed. If you look at the natural body posture of a mouse when it's exploring its environment, you'll see that their heads are tilted down to allow them to whisk the floor. So we actually head fix our animals in this 35-degree angled clamp. We've noticed this actually has a profound effect on how well they move around while head fixed. And they appear much more comfortable being able to whisk the base of the maze as they run through, and as well as being able to sit and groom in a more naturalistic position. Another issue that we had was that the mice were reacting quite adversely to the light that we use during recording sessions. So we need to use this light so that we can accurately place a pactoral electrode in the cranial window. Of course, we're doing this a number of times during the recording session. And if you look at the animal's activity while this is happening, they become very active while the light is on, essentially because they're running to escape the light. And then after it's switched off, usually when we're actually having a cell recording, they tend to show very low activity. And with some, they even exhibit freezing behaviors, which is unsurprising, but somewhat problematic. So we came up with a very simple solution, which was develop a visor that you can see here, which we attach to the head plate. And essentially this blocks much of the light from above whilst leaving the visual field in front of the mouse free. And what we found is that whilst they're wearing this visor, the animals show a much more stable activity profile during the changing light conditions in a recording session, and show less indication of stress throughout. So what I've hoped that this demonstrates by sharing a couple of these issues is how small changes to try and reduce the stress in head-fixed animals can really help get the most out of the recording session in terms of the animal's behaviour. So what kind of behaviours have we been looking at? Well, first we wanted to just track the animals while they navigate around an environment where they're rewarded for visiting a particular location, and see how well we could measure the changes in their behaviour when this information about the reward location is changed. So in the first day, the animals are placed in this T-maze style arena where they have to run up the central corridor and make either a left or right turn. They then pass through a door and return to the start position. And the first day, they're awarded in both left and right arms. Here, they're zones 7 and 3. And on the second day, the reward delivery only occurs in one of the two reward locations. And you can see that both from the heat map and from the proportion of turns that they make into the rewarded arm and the time that they spend in that part of the maze, that it's very easy to see these mice developing a preference for the reward location. More interesting, however, is that when you switch the reward location to the previously unrewarded zone, you can see a reversal in the animal's behaviour, and they switch their preference to the newly rewarded location. And once again, when the reward location reverses back to the original place. And this is good to see because in my experience, mice in particular can be pretty inflexible and can quite easily develop spatial biases that are difficult to train out of. Whereas these mice are showing relatively quick shifts in their behaviour that we can see both qualitatively and quantitatively. And we're going to develop this task further into a probabilistic reversal learning task suitable for head-fixed mice in the mobile HomeCage. But for the rest of the talk, we're going to be talking about another behavioural task that we've been using, this cued goal-localisation task, which came about mainly because I wanted to see how well these head-fixed mice would learn some kind of cued response in the mobile HomeCage. So in this task, they were required to navigate around a track containing both tactile and visual cues. I've trained them in both the circular track as well as the T-maze that I showed in the previous slide. And they had to locate a target zone, which was marked out by a square of sandpaper on the base of the maze. And this time they would actually have to sit and wait in this relatively small area for 2 seconds before the reward was delivered. And if they ran straight over, or if they didn't complete at least half a lap before returning to the target zone, they didn't get the reward. And you can see that for the mice that I trained to do this, it took them a couple of weeks to reach about 70–75% accuracy. And this video shows the animal doing this quite nicely. He runs around, stops, in the correct place and then runs around again. And you can also see that over time, they show a reduced tendency to stop in other non-target locations, whilst their overall activity remains pretty consistent across the sessions. So this tells us that in a fairly short space of time, these head-fixed animals are able to effectively learn to respond appropriately to a cue in order to obtain reward. And we're using behavioural tasks like this to form the basis of our experiments, investigating the neural mechanisms underlying spatial navigation and memory. So it's at this point that I'll hand over to my colleague, Jon, who's going to talk more about the process of getting intracellular recordings in the mobile HomeCage in this task.
Thanks, Sarah. I'm Jon Palacios and will tell you how we are using Neurotar's mobile HomeCage to perform in vivo whole cell patch clamp recordings. To understand how behavioral experiences alter activity-induced synaptic plasticity, we must measure subthreshold membrane dynamics, which are shaped by synaptic inputs and suprathreshold firing of the neurons. To this end, we perform whole cell patch clamp experiments in hippocampal CA1 neurons in awake mice, which have been trained in a acute goal localization task, and hence they are running in a familiar environment. We chose to record from hippocampal CA1 neurons due to this brain structure has been linked with declarative memory formation, spatial memory, and contextual memory. In addition, behavioral tasks that my colleague Sarah has just presented are meant to be hippocampal-dependent tasks. We perform a cranial window of around 1 millimeter diameter above each hippocampal CA1 areas the day before, so the mouse has enough time to recover from the surgery. We start by introducing a patch pipette through the cortex until it reaches close to the hippocampus. Due to the head position, which is tilted 35 degrees, we compensate the shift to target CA1 area of the hippocampus. To find a CA1 neurons, we go ahead in 2-micron steps. Whenever the pipette encounters any cellular membrane, the resistance will increase, which informs us to inject negative pressure and hyperpolarize the cell when the patch pipette reaches 100 megaohms. Lastly, if the resistance reaches until 1 to 10 gigaohms, will mean that the seal has been set, and further negative pressure will open the formed cell, giving access to the submembrane dynamics. At the end of the experiment, we perfuse fix the mouse to post hoc double check that we were recording from CA1 area of the hippocampus. On the top left, you can see an example of a whole cell patch clamp recording on current clamp mode, so we are able to see the spiking activity of the neuron. Below is the path followed by the mouse while the recording lasts, highlighting with open red circles the position where the action potentials happen. In this case, the mouse made around 15 laps, and its movement is quantified just below. The blue line represents the distance moved over time, and the red line is the cumulative distance moved. One lap in this 32 centimeters arena is around 1 meter long. So, as you can see, the mouse explored a cumulative distance of 15 meters. On the middle of the slide, you can see the interspike interval, which shows a binomial distribution, where some spikes occur at short intervals and others with higher temporal distance between them. This tells us that there is some clustering of the spikes. Below, you can see the angular position of the mouse with respect to the mobile HomeCage, which has been color-coded with the animal's speed. Hence, an horizontal trace on the plot will be color-coded in dark blue, since the mouse is not moving. When the mouse moves, you can appreciate lighter colors. In the same plot, open red circles represent spikes occurring during exploration, which were obtained when the mouse was moving at higher speed than 2.5 centimeters per second. On the top right-hand side, there is a raster plot to visualize where do spikes occur with respect to the mobile HomeCage. Here, each line represents 1 lap for the animal's movement. Below, we quantify the spike number in 30-degree sector bins in blue bars and time spent in each sector by a red line. Thus, on the bottom, we see the spike frequency normalization to the time spent in each sector. As it can be seen, spikes are not regularly distributed, but at some locations within the mobile HomeCage arena, there is a higher spiking probability. The fact that neurons fire more action potential at the specific locations is a hallmark for hippocampal CA1 place cells. 70% of neurons in CA1 area of the dorsal hippocampus behave as place cells, meaning that specific neurons fire action potentials regularly whenever the animal crosses a particular field. Thus, place cells allow the formation of a precise cognitive map that can inform the animal on its location at any time. To address whether a spike in activity of recorded neurons encode spatial information content, we plot the spike frequency according to animals' angular position in the mobile HomeCage. Here we have analyzed recording where the mouse made at least 5 laps and evaluated only exploratory periods. On the top, you can see up to 7 examples where recorded spike showed certain degree of preference regarding the animal's position. However, not all the neurons seems to encode spatial information content, since their action potentials were regularly distributed on the behavior arena. As mentioned before, action potentials represent the output of single neurons. To analyze the pattern of action potential output, we classified spike output in 3 different groups. First, we have outputs that arise as a single action potential. Second, we have bursts of action potentials which are composed between 2 to 4 spikes. And third, action potential bursts composed of 5 spikes or more. To make this classification, we define a burst if 2 or more action potentials happen in a time window of less than 100 milliseconds, one to each other. To make this action potential classification, we follow the rationale that in freely moving mice, hippocampal CA1 spike outputs occur in single, duplets, or triplets regularly. However, some spike bursts are composed up to 5 to 10 action potentials. This superburst has a higher probability to transfer encode information. We have also analyzed if this classification into 3 groups differ in epochs where the mouse is still or in repose, and those epochs where the mouse is freely exploring. Importantly, to understand neuronal output, we must understand synaptic input that trigger spike generation. From a whole cell patch clamp experiment in current clamp mode, we can infer that submembrane dynamics are the result of synaptic inputs and its relationship with internal properties of the neuron. On the middle panel, you can see a magnification of the upper trace to highlight that membrane potential depolarization might give rise to action potentials. Interestingly, not all the membrane potential depolarization epochs give rise to action potential, but this might depend on many factors, such as degree of depolarization or spiking threshold between others. To further analyze this, we define a membrane depolarization epoch whenever there was an increase of 10% over membrane potential baseline. If we zoom in more, as shown in the bottom panel, we could even appreciate single fast events, presumably synaptic inputs, which produce these membrane potential depolarizations. In the top panel, you can see now that the frequency of these events is much higher than the occurrence of action potentials, and our preliminary analysis shows that they are theta frequency modulated. These submembrane dynamics that give rise to action potentials are analyzed in the right-hand side of the slide. To this end, we focus on the 100 milliseconds that preceded each isolated action potential and quantify the development of membrane potential until the occurrence of the spike. Our preliminary analysis shows that single occurring action potential are preceded by regular steps that give rise to the generation of a spike, while bursts of action potential occur in more abrupt changes of the membrane potential. We are further analyzing this data to establish if more coordinated synaptic inputs give rise to different patterns of development of submembrane dynamics. In addition, we are also interested in how synaptic plasticity might change input-output relationship in individual neurons and locate key time points in the behavioral task where synaptic inputs are transformed into spike outputs more efficiently. That's for me. I'm now going to pass things back to Liam for an audience poll. Thank you.
Hello, here speaks Alexander Didiatiev. Our presentation is about imaging of microglia. And first, let me give a brief introduction to microglial cells. These are immune cells resident in the central nervous system. They are broadly distributed throughout the brain, spinal cord, and retina. The cells have characteristic ramified appearance in the normal CNS tissue with numerous fine processes that do constant surveillance of the tissue environment. Microglial cells have a capacity to rapidly transform to alerted, activated, and fully reactive states in response to stress, infection, or any type of tissue damage. This transformation is important for tissue maintenance, protection, and restoration. Moreover, microglia contributes to guiding and assisting in engagement of adaptive immune response. As we are synaptic physiologists, for us the most fascinating aspect of microglia activity is the interaction with synapses, so which may result either a synaptic loss, so-called synaptic stripping, or stimulation of spinogenesis by molecules released from microglia, such as the brain-derived neurotrophic factor BDNF. We think that it would be very important to put these new findings in the context of the tetrapartite model of chemical synapses, which considers exchange of molecular signals between the presynapse, postsynapse, astroglia, and perisynaptic extracellular matrix. In this slide, you can see that microglial processes approach synapses and establish transient contacts with all synaptic components, including presynaptic boutons and postsynaptic spines. Some molecules, like ATP, released by neural cells, may attract the microglia, and others, like C1q, eventually provide a tag to instruct microglia to attack a synapse, while other factors may repel microglia. This is a fascinating new field, and obviously it remains to be learned a lot about this pentapartite stage of synaptic dynamics. To understand factors controlling microglia behavior, we like to image microglia. How to do this properly? How to gather reliable information that would reflect predominantly real microglial properties rather than specifics of our observation method? Shall we use anesthetics or do imaging in awake mice? At which time after surgery? These are important methodological questions, particularly considering the results of a recent electrophysiological study, which demonstrated that application of isoflurane, which is one of most commonly used anesthetics, so may substantially block the microglial response to ATP, which is known to be essential for guidance of microglia. Also, isoflurane depolarize microglial cells in vitro. So, hence, the aim of our study was to compare microglia surveillance and damage-related motility in awake mice versus mice anesthetized by ketamine or isoflurane. So, moreover, we compared microglial activity in acute versus chronic preparations. The methodology used and the results obtained will be presented by my colleague Bailun soon.
From here, I'm going to introduce the details and the results of our study. So in this study, we implanted a glass cranial window, which allowed us to perform in vivo two-photo imaging of the same animals during several months. Briefly, after the animal was anesthetized by isoflurane under the head fixation, A 5 mm diameter cranial window was gently made over retrospinal cortex and then covered with a 6 mm glass cover slip. A custom-built head plate, as shown here, was then implanted with superglue and dental cement. For awake condition, mice were head fixed under the 2-photon microscope using a mobile home cage in which the animals could freely move a light carbon cage around them. To address the differences of microglia morphology and dynamics between awake and anesthetized conditions, we performed the same imaging procedures in awake, insufferin, and ketamine anesthetized conditions respectively. The imaging area was located in the retrospinal cortex. That is one of the cortical areas critically involved in spatial navigation, which is affected at very early stages of Alzheimer's disease. The imaging schedule is shown here. 2-photon imaging was performed first in acute preparation on the first and second days after the window implantation, and then in chronic preparation, that is 4 months after the implantation. The longitudinal design allowed a within-animal comparison of microglia morphology and motility in awake versus anesthetized conditions on the same day, with a 2-hour interval between sessions. And each session included imaging of microglia at the surveillance state and after photo damage induced by laser ablation of a single microcell at 2 separated sites. Here is an example of microglia at the surveillance state, and as you can see, microglial processes are highly dynamic, surveilling the surrounding environment. However, after photo damage, microglial processes are rapidly approaching to the center of the damage. These data were collected as Z-stacks with images acquired at 20-second interval for 33 minutes and in total for 110 frames. The lower panels demonstrate 2-dimensional tracing of microglia processes with a 30-minute interval in the surveillance state and microglia response to a photo damage. To categorize microglia at the surveillance state, we trace the processes and compare the length and number of primary microglia processes and the total length and number of terminals of all processes per cell, as shown here. In acute preparation, during insufferin anesthesia on day 1, the average length of microglia primary processes was significantly elongated compared to awake condition. In contrast, during ketamine anesthesia on day 2, the average length of processes was unchanged, but the number of total processes was increased compared to awake condition. Additionally, both the average length of primary processes and total length of all processes on day 2 in awake condition increased significantly compared to day 1. All these data indicate that anesthesia as well as time interval after window implantation differently affect microglia processes in acute preparation. In chronic preparation, we apply the same analysis, and in line with acute experiments, we observed an increase in the length of primary processes in insufferin anesthetized condition compared to awake condition, but no difference in other structural parameters and motility was detected, which indicates that insufferin significantly affected the length of microglia primary processes, while ketamine had no effect on the length and number of processes in chronic experiments. For the analysis of microglia at the surveillance state, we traced each process twice with 30-minute interval, and the changes were categorized as disappear, decrease, no change, increase, and appear, as shown here. We only found a difference in acute experiments with fewer primary processes disappearing and more primary processes decreasing under insufferin anesthesia than in awake mice on day 1, while the appearance and elongation of new processes were not affected. This indicates insufferent anesthesia affected the turnover of microglia processes. Analysis of acute versus chronic preparation revealed the effect of an interaction between condition and preparation on the total length of all processes. We observed an increase in the total length of all processes in chronic preparation compared to acute preparation in the first awake condition, as well as in insufferent and ketamine conditions, suggesting that in the chronic preparation, microglia are more in the surveilling rather than activated state as compared to the acute preparation. To study the damage-directed microglia response, we induced the photoaggregation of a single microglia cell. As you can see, the site of damage remained visible as a fluorescent spot, to which other microglia cells extended their processes, forming a ring-like structure. And Shaw analysis was used to calculate the velocity of damage-directed process motility. The precise frontier was measured by detecting the peak intensity of microglia processes on the circumference at a distance from the injury spot. The distance of peak intensity was plotted a long time, and the linear regression was fed to the observed data. Its slope was used to calculate velocity. The quantification of microglia process velocity shows that in acute experiments, there was no effect of anesthesia on velocity, but there was a significant difference between day 1 and day 2 under awake conditions. And in chronic experiments, the velocity was increased under insufferent compared to awake condition on day 1. Direct comparison between acute and chronic preparations revealed that velocity was higher in insufferent condition in chronic preparations. Taken together, the directed motility of microglia to a photodamaged site is strongly affected by preparation type and insufferent anesthesia. Putting together all data, we conclude that the insufferent anesthesia increased the length of surveilling microglia processes, both in acute and chronic preparations, while ketamine increased the number of microglia branches in acute preparation only. In chronic but not in acute preparation, the extension of microglia processes towards the damage was faster under insufferent anesthesia than in awake condition. Our conclusion for this study first reviews potentiating effects of insufferent on the length of surveilling microglia processes and microglia response to a damage. Therefore, we recommend to use awake mice with a chronically implanted transcranial window for the studies of microglia morphology and function in vivo. However, if imaging in awake mice is not feasible, then ketamine anesthesia in chronic preparation is preferable to insufferent. Finally, we want to thank all our colleagues and collaborators involved in this study for their valuable contributions, and we also appreciate the support from the Second Young Glia program. At last, thank you very much for your attention. And now I'm going to pass to Liam for the audience poll.
Welcome everyone. My name is Norbert Hájos and I'm leading a research group at the Institute of Experimental Medicine, Hungarian Academy of Sciences in Budapest. Our research group is interested in neuronal circuits and their operation. We focus our studies predominantly on the amygdala. Amygdala is a complex structure that is located deep in the brain. One of many amygdala nuclei we study most, this is the two main regions, the so-called basolateral amygdala complex and the central amygdala. As shown in the light microscopic image on the right-hand side, taken from the brain of a mouse, in which all GABAergic cell bodies are green, you may see that the basolateral amygdala contains as many GABAergic neurons as the surrounding cortical regions, like, for instance, the piriform cortex. Thus, this amygdala region is a part of the cortical structures. In contrast, the vast majority of neurons in the central amygdala has a GABAergic phenotype and have similar features as the median spiny neurons in the striatum. Therefore, this central amygdala is closely related to the striatal structures. Amygdala is involved in various brain functions, including memory formation, decision-making, emotional responses, social interactions, and even food intake. Our overall aim is to reveal the information flow within the amygdala complex during aversive and appetitive behaviors that are known to be processed by this brain region. To this end, we record spiking activity of individual neurons in different regions of the amygdala in head-fixed behaving mice. Specifically, our first goal is to establish conditions that allow us to study the behavior of head-fixed mice. Our second goal is to perform recordings of a spiking activity of a well-isolated single neurons in the amygdala region in behaving mice. So what are the methods that we are using? In line with our research goals, we aim to use a setup that is additional to providing the recording stability of head-fixed conditions, offer behavior capabilities comparable to freely moving recordings. That's one of the key issues. We perform our behavior experiments in awake head-fixed mice placed in mobile home cage, and to conduct monitoring of neuronal activities in awake head-fixed mice, we obtain unit recordings by silicon probe and juxtacellular record electrodes in mice. So, first, let me show you an example for studying aversive memory processes using a mobile home cage. Previous study has established that amygdala is critically involved in cue-dependent fear learning. So, therefore, we habituated mice in the mobile home cage for 7 days before fear conditioning. Then we placed the mice individually into a fear conditioning box where a tone, a conditioned stimulus, was paired with a mindful shock, which is an unconditioned stimulus. And this has been repeated by several times. During this fear acquisition, the majority of mice, like here mouse number 2, here, gradually develops so-called freeze behavior in time as more CSUS pairing are presented. Such pairings are indicated with dashed line on this graph. In contrast, few mice, like for instance the mouse number 3, show only moderate freezing response during the conditioning. On the day following the fear conditioning, we place back the mice into the mobile home cage for testing their fear memory. After a baseline period, we presented the tone 4 times for each mice. Here I show data obtaining from 3 mice. 2 of them showed a marked conditional response, freezing, during the tone presentation. Importantly, testing the fear memory recall for 2 more days, these 2 mice showed a still significant freezing behavior when the tone was presented, but the baseline immobility of mice on the day 3 was reduced, which may indicate actually that the gradual decreasing of the anxiety level. The 3rd mice, which didn't show a normal fear acquisition, as I have shown previously, did not show freezing behavior under this condition either. In summary, these data show that, like in conventional use apparatus for fear conditioning, fear memory can be studied by using the mobile home cage. Next, I would show an example for motivationally driven behavior. In this case, we train mice to run for longer time in the mobile home cage by motivating them with sucrose. Via a liquor delivery system, we give a drop of sucrose solution when the mouse is willing to run more. This graph shows in the middle that the mouse almost doubled the time of running within 6 days of training. Here I want to show you a video taken from a well-trained mouse which learned to run to complete 30. Here he got a drop of sucrose, and after, at the very same place, he stops. And again, when he received another drop of sucrose, he performed another running. So these data show that collectively that actually the mobile home cage is good to study behavior phenotype. The last part of this presentation, let me show you 2 examples for recording of neuronal activity while mice are moving in the mobile home cage. In this first example, silicon probe was used to detect the spiking of neurons. We acutely inserted the silicon probe in this case into the central amygdala. And after 30 minutes of waiting period, we could record neuronal spiking for more than a tenth of minutes. Here I want to show a short video demonstrate that the recording of unit at the deepest contact site was stable when the animal was moving in the mobile home cage. So you can see the animal is moving, and when the animal stops, the unit recording are actually stable. The second example shows a juxtacellular recording of neural activity in behaving mice. Here we use a glass pipette filled with a sodium chloride solution containing a tracer molecule, neurobiotin. If we are close to the neuron, spiking activity can be clearly monitored. After recording, we modulate the spiking of the neuron by current pulses, which allows the neurobiotin to get to the neuron. After 5-10 minutes of electroporation of the tracer molecules, we can fix the brain and visualize the neuron that was recorded. Here I would like to show an example when a spiking of neuron was obtained juxtacellularly while the mouse is moving in the mobile home cage. Here on the left-hand side, you might see the recording and the spiking activity of a single neuron while the mouse is moving. And when the mouse stops now, then we have the cell and the spiking activity can be clearly seen. These examples collectively show the spiking activity of neurons can be obtained in such a deep structure as the amygdala in mice behaving in the mobile home cage. Now, let me conclude. Mobile home cage is a platform for studying neuronal activity in a head-fixed configuration, but the mouse is engaged in affective behaviors. Both field memory processes and the motivationally driven behavior can be studied in mobile home cage. And using either silicon probes or juxtacellular positioned glass electrodes, stable recordings of spiking activity of individual neurons can be reliably obtained in head-fixed behaving mice using the mobile home cage. And last but not least, I'd like to acknowledge, first of all, Neurotar Ltd, provide a prototype of a liquid reward delivery system. And here down, you can see the funding body who supported our work. Thank you very much for your listening.
All right, thanks Norbert for the great presentation. Moving on to the Q&A, we had a number of questions come in during registration and some during the live session, so let's jump right in. First question today is, how long does it take to train a mouse in the mobile home cage, and are there any critical factors that have to be taken into account? And so I'd assume that depending on your research, you could have a different answer, but why don't we start with you, Norbert?
Yep. First, we habituate the mice for 3 days in the mobile home cage, usually twice a day for 10 minutes. Then we start the training. Training takes usually 7-10 days. And we have found that mice learn faster if their whiskers can touch time to time the wall of the mobile home cage platform. So we think this is a critical factor, the touching the wall by the whiskers. Okay.
Okay, thanks, Norbert. And Sarah, I know you and Jon covered this a bit in your presentation, but could you just give us a bit of a recap of your experience with regards to habituation and maybe other critical factors to consider?
So if we're just talking about getting the mice to run comfortably around the mobile home cage, I'd say that we see them doing that within just a couple of short sessions of head fixation. But factors that I would think are quite important in helping them do this, we give them running wheels in their cages, make sure that the mice are well handled before the training starts. We habituate them to the cloth wrapping technique that we use to get them into the head fixation clamp. And we also tend to give them time in the mobile home cage maze itself whilst they're freely moving, so that they're in a familiar environment once they're finally head fixed for the first time.
Okay, great. And just passing the question off to Waylud and Alexander, what's your experience with this?
Well, this is a very practical question. So in our case, actually, we didn't use pre-training in this particular study because there was no behavioral task involved in our study. And we did have these 3 sessions of habituation. Each habituation was 10 minutes, and after the habituation, we didn't see the animal to be stressed, so this was working for our case.
Oh, okay, perfect. And so this next question is for Jon. How easily can you expect to find CA1 pyramidal neurons with place cell activity?
Yes, from our experience and the few single cell recordings that we have, I say few because altogether they are not more than 20 recordings. We found a wealth of neurons that show a location preference to a spike. However, we are planning to do acute recordings with silicon proofs where we expect to record more than 100 or 200 units per experiment, and then we will have definitive results.
Perfect. Thanks, Jon. And so we have a question here about the mobile home cage and customization. How much can the MHC and tracking system be customized to make it suitable to reproduce a more complex environment? Jon, I think you know a bit about this one. What do you think?
Up to now, the mobile home cage is limited to circular mazes, but Neurotar provides custom design mazes. Regarding the tracking system, the mobile home cage has 4 digital TTL pulses that are triggered when the animal stays in a certain zones within the arena. Then it's up to the users how to play with those pulses to trigger more elaborated responses, such as sound cues, visual cues, reward, or punishment.
Excellent. All right. So this next question is for Sarah. So Sarah, how well did head-fixed mice learn in the mobile home cage compared to either freely moving animals or other head-fixed setups?
So for the task that I showed in my talk, where the mice had to go to a specific target location and wait in order to get the reward, it took them about 2 weeks training to get them to do that in the mobile home cage. Whereas in a freely moving animal, they'd probably be able to do that in just a few sessions, I would expect. But I think that's kind of typical with any head-fixed setup, that it takes longer for them to learn than you'd expect in a freely moving animal. But in a lot of the tasks that I've been running, I tend to follow similar training protocols as those that have been used in the virtual reality systems. And I've found that in some cases they learn in fewer sessions by comparison. And I think a key factor with that is that in the mobile home cage, the mice are able to rely both on the tactile as well as the visual cues in their environment. One thing that we're looking into now is whether training the animals in that task freely moving first, so before they undergo any surgery and then head fixation, whether that information will translate well enough such that they don't require as many training sessions in head fixation for them to be able to perform the same task in the mobile home cage, which should help with training.
Okay, great. Thanks, Sarah. And so we have a question here about juxtacellular electrodes. So I'm going to direct this one to Norbert. What is the average time for recording with juxtacellular electrodes in head-fixed behaving mice?
So we found that the average time for a juxta recording is about 10-15 minutes. And this depends on many factors like the size and the quality of the craniotomy and the tip of the glass pipette. We have recorded in some occasions neuron spiking for more than 20 minutes while the mouse was grooming, running, receiving several air puffs. So that's, if everything goes well, a significant data can be collected during various behaviors.
Okay, perfect. That's a great answer. So the next question I think is most relevant to Wei-Lun or Alexander. Can you specify how image analysis was performed to quantify morphology of microglia and velocity of microglia approach to a photo damage site?
Well, we use the Fiji package for the analysis. And basically, in the surveillance state, we trace the processes by the Fiji plugin called Simple Neural Tracer. And then we further confirmed by manual track. The same cells were traced twice at time 0 and 30 minutes to quantify the cell motility. Well, regarding the damage-directed state, we used the Shaw analysis, which also implanted in Fiji package to calculate the velocity of processes approaching to the center of the damage. This is basically how we did the analysis.
All right, thanks, Wei-Lun. And so the next question would be for you, Norbert. How long do you wait after insertion of a silicon probe into the amygdala region before the recording? And what is the stability of units on an acutely inserted probe?
So after insertion of a silicon probe, we usually wait about 30 minutes, but often an hour. So we have found that the stability of the unit upon acute insertion of a silicon probe is usually tens of minutes, so at least half an hour. So for that long, we can really recall stable units. But if we are losing units, we can move slowly deeper and to find and search for new units and to record a distinct neuronal populational activity. Thank you.
Fantastic. Thanks, Norbert. So this next question deals with behaviors and maybe the limitations of head fixation, but to what extent in the mobile home cage are behaviors like grooming, sniffing, and sleep affected?
So with the sniffing, I think the angled clamp that we use definitely provides a huge improvement in terms of allowing the animal to sniff and whisk the environment as if they would in freely moving. With the grooming behaviours, the mice are able to carry out face grooming, but because of the head fixation clamp, they're unable to do a full body groom while they're in the mobile home cage. But what we've noticed is that on most occasions they'll carry out a full body groom as soon as they're released from the clamp. We haven't looked at sleep specifically in the mobile home cage. All our training and recording sessions are carried out during the animal's active phase, so it's not something that we'd expect to see during our studies. But I would say that for anyone wanting to look at sleep in the mobile home cage, something important to consider would be the rate of airflow underneath the cage. So you want the home cage to float easily enough so that the mouse can run around with minimal effort, but it also then needs to be stable enough underneath them so that when they're not moving, it's not wobbling about, and it should allow them to sleep, maybe.
All right, perfect. Thanks, Sarah. And in the interest of time, we're just going to make this next question the last one. We received a few questions during registration asking about the challenges of making measurements in moving animals, which John said that he could address here. So, John, how do you mitigate motion artifacts while recording? And how long can you keep a whole cell recording while an animal is actively moving?
Regarding the motion artifacts, I would recommend having a very good reference electrode, which is stable upon movement. In addition, if the mouse touches any metal lever or electricity conductive metal, you might expect to see a small artifact. These artifacts are easy to remove because they have a very characteristic electrophysiological signature. Regarding how long can you keep a seal, I would say that it depends in several factors. First, you have the stability of the clamp, of course, which is quite good. However, stability also depends in the size of the craniotomy, since you might see brain movement due to breathing and heart pulsing. This can be minimized if you put some solid agarose plaque to stabilize the brain and the recording electrode. Secondly, stability will also depend on how much the mouse is moving and if those movements occur smoothly. That's why it's always recommended to train the mice beforehand to avoid any abrupt movements. Overall, I would say that a good 10 minutes can be expected from a trained mouse, but this is very variable and might go from 5 to 20 minutes.